To get started, I'm going to immediately pass it over to Dr. Klopp to introduce our speaker for today. And thank you all for coming.
Hey everybody, so today's speaker is Yuichi Fukutomi. He did his PhD at the University of Hokkaido with Sugiyuki Koshikawa working on Drosophila pigmentation. And then he went to Ayata Kasashi's lab at Tokyo Metropolitan University to work on Drosophila pigmentation. And then he came to our lab as a postdoc to work on, you guessed it, Drosophila pigmentation. I swear that was not the original intention. He was planning to work on something completely different, but I can vouch from my own experience that Drosophila pigmentation is addictive because there are many different reasons to work on it. You can use it as a model for pattern formation or the evolution of gene regulation or convergent evolution, just about anything else. And in Yuichi's case, he's been using it as a model to work on the evolution of dominance, the evolution of dominance relationship between alleles. And so that is one of the many things that he talked about.
Thank you for introducing me. And I'm going to start my presentation. So it's about the connection between the evolution of dominance and plasticity. And I'm working on fly pigmentation. So, well, genotype-phenotype map is the relationship between the genotype and the phenotype. And it's important in terms of evolutionary biology because the fitness for each genotype can be determined. So when the shape of genotype-phenotype map changes, the fitness for each genotype can be altered. So what determines the shape of genotype-phenotype map? So one of the phenomena is the additive effect from each allele. And next one is the allelic dominance. When there's allelic dominance between the alleles, the shape of genotype-phenotype map will be different. And the next one, if the phenotype is determined by multiple loci, so there can be epistatic effects, and the genotype-phenotype map will be different. So in the case of additive effects, so each genotype can have the different phenotype. And on the other hand, in the case of allelic dominance, so multiple genotypes can create single phenotype or similar phenotype. And in the case of epistasis, a single genotype at a certain locus can have the multiple phenotypes depending on the genotype on the another locus. So, well, a single genotype at a certain locus can have multiple phenotypes. Other than the case of the genetic effect by another locus, it's possible for certain genotype at a certain locus can have multiple phenotypes. So that is the case for phenotypic plasticity. So in the case of phenotypic plasticity, a single genotype can create multiple phenotypes depending on the environmental conditions. And when the allelic dominance and phenotypic plasticity are compared, so the relationship between the genotype and the phenotype sounds like reversed relationship because the allelic dominance for allelic dominance, the multiple genotypes can create a single phenotype. On the other hand, the phenotypic plasticity, single genotype can create multiple phenotypes. So however, so they can stem from the same developmental mechanism. So for example, a threshold-like property of development can be considered. So a determinant for the phenotype, such as gene expression level or hormone titer, can change linearly depending on genotype or environment. And if there's a threshold and the determinant is below the threshold, so this system will have a certain phenotype. And on the other hand, the determinant is above the threshold, so it can have the alternative phenotype. So Well, so this is the threshold-like property of development, and it means that two phenotype flips depending on the threshold of a certain determinant for phenotype. So the threshold-like property of development can explain phenotypic plasticity. So here's the empirical example of the phenotypic plasticity. So it's the case of horn length of dung beetles. So the dung beetles have the two discrete morphs of horns, the large horns and short horns. And so it depends on the nutrition level. So let me grab the pointer, I'm sorry. So sorry. So the horn length is depending on nutrition level and the amount of juvenile hormone. And the juvenile hormone titer decreases when the nutrition level goes up. And so when the juvenile hormone titer is above the threshold, the horn will be shorter, and the juvenile hormone titer is lower than the threshold, the horn will be longer. And on the other hand, for the case of the allelic dominance, a theoretical model predicts that the dominance can evolve through threshold-like property of development. So evolution of dominance is the phenomenon where the dominance relationship between alleles differ between species, populations, or sexes. So let's assume the case where the dominance is reversed between species. For example, in one species, there are two alleles, large A and small a, and large A is dominant. And in another species, the allelic dominance is reversed. And let's assume the case where the phenotype is depending on the gene expression level, and if the gene expression is below the threshold, a certain phenotype can be observed, and the alternative discrete phenotype can be observed when the gene expression level is above the threshold. So if two alleles have the different expression level, it's possible for two homozygotes to have the discrete different phenotypes. And in the case of species 1, the heterozygotes will have the intermediate expression level. And if the expression level is above the threshold, it's possible for these two genotypes to have the same phenotype. On the other hand, in another species, so even though the expression level is totally same as the another species, if the threshold is higher than species 1, it's possible for the heterozygotes to have the same phenotype as the homozygotes with small a homozygotes. So, wow, so I explained that the threshold-like property of development can explain both of the phenotypic plasticity and the allelic dominance. So based on that, so it's possible to hypothesize that the allelic dominance and phenotypic plasticity are directly connected in the same developmental system. So I examined this hypothesis by using the female limited color dimorphism in Drosophila montim clade. So in this clade, it's possible to observe the abdominal pigmentation, and males have the monomorphic pigmentation. On the other hand, females have the dimorphic pigmentation, light phenotype and dark phenotype. And they will show the Mendelian inheritance, and there are light allele and dark allele. So the light homozygotes show the light phenotype, and the dark homozygotes show the dark phenotype. And then the phenotype of the heterozygotes were examined, And most of the species, the heterozygotes will show the dark phenotype. On the other hand, some species show the light phenotype. So it means that most species show the dominance of the dark allele, but some species show the dominance of the light allele. And then in the previous research, locus associated with the dimorphism was examined. And so it turned out to be PDM3 gene. It's a transcription factor to repress melanin pigmentation. But so far, the species examined was all of them were dark dominant species. But we don't know anything about the light dominant species. So what happens in the light dominant species? So there should be two hypotheses. So number one is the light dominant species will have the different genetic basis. So it means that the locus other than PDM3 is responsible. And hypothesis number two is it's PDM3 and the dominance reversal is happening in the light dominant species. So I investigated the hypothesis by using GWAS analysis or selective genotyping. And for GWAS analysis, what I did is the K-mer-based GWAS analysis. At first, I extracted DNA from dark females and light females and sequenced the genome. And so in computation, we chopped the sequence trees into K-mer, and then we counted the K-mer frequencies. And after that, we found K-mers associated with the phenotype and assembled them. So it means that the region associated with the dimorphism was identified in the nucleotide. So after doing the GWAS analysis and the selective genotyping, so what we found is the PDM3 locus was associated with the phenotype in both the dark dominant species and the light dominant species. And we found that the intronic mutations in PDM3 are associated with the dimorphism. So the answer is the hypothesis 2. So it's PDM3 and the dominance reversal is happening in the light dominant species. So, and then I investigated the relationship between dominance and plasticity. So, so you might think that, wait for a second, why it's possible to, examine the relationship between dominance and plasticity in the fly abdomen, big fly abdomen. So actually the fly abdomen pigmentation has been studied for phenotypic plasticity. And for example, in Drosophila melanogaster, so this is a monomorphic light species. And when they are reared at the higher temperature, it shows the light phenotype. And it looks like similar to the light phenotype in Montium clade. And when they are reared under lower temperature, so it looks like the pigmentation is much darker, and it looks like a dark female in the Montium clade. So in this clade, the difference is made by the genetic difference, and so in this case, the phenotypic difference is created by the environmental effect. But so in this clade, the effect of temperature was not examined. So we investigated the phenotypic response to temperature. And then, so we used these 4 species, two dark dominant species and 2 light dominant species. So at first, in both of the dark dominant species and light dominant species, so I investigated the phenotype of dark homozygotes. So this is the distribution of the phenotype. And so let me simplify by lining up the average like this. So, okay, so, well, this data shows that dark homozygotes had the little plasticity in the body color. And I also examined the light homozygotes. And so light homozygotes also had the little plasticity in the body color. So that tendency was observed both in dark dominant species and in light dominant species. And then we investigated the phenotype of the heterozygotes. In dark dominant species, the body color was almost dark. So it means that the dark PDM3 allele is fully dominant regardless of the temperature in dark dominant species. So then we investigated the phenotype of the heterozygotes in light dominant species. And it shows that the phenotype had the phenotypic plasticity. So in the case of Bocchetti, the dark allele was dominant at the lower temperature, but the light allele became dominant at the higher temperature. And in the case of Jambolina, the two alleles were co-dominant at the lower temperature, but the light allele was dominant at the higher temperature. So in both species, the light PDM3 allele became increasingly dominant at the higher temperature. So taken the results together, so dominance reversal of PDM3 alleles is directly connected with the evolution of thermal plasticity. So this is the key concept for subsequent projects of mine. So after this, I'm going to present about two projects. So number one is what is the developmental mechanism to connect the dominance reversal and the evolution of phenotypic plasticity. So number two is the interspecific dominance reversal due to the evolution of Fischer's modifier. So for the number one, so what is the developmental mechanism to connect them? So this is actually an ongoing project, and I'm going to propose some kind of model or hypothesis based on my current data. So first of all, to understand the mechanism to connect dominance and plasticity, so it's necessary to understand what is the developmental mechanism to determine the phenotype. So PDM3 is a replacer of melanin, and so when it's knocked down in the midline of Drosophila melanogaster, so it means that the PDM3 expression level is decreased. The pigmentation will be expanded compared to control mice. And then when the PDM3 is overexpressed in the posterior region of abdomen, so it means that the increase of the expression level, so it will lead the fly to lose the pigmentation in the posterior abdomen. So based on that fact, I had the hypothesis to explain the phenotypic differences between the dark fly and light fly. So the hypothesis one, PDM3 is not expressed at the place of pigmentation, and it's expressed at the place where the light phenotype is observed. And there should be alternative hypothesis. So the dark phenotype at the serrata is created by another gene, and in the light fly, the dark pigmentation is repressed by the expression of PDM3. So both two hypotheses assume that there is a spatial difference in the expression of PDM3. So I examined the hypothesis by antibody staining and in situ hybridization. So what I found is there's no obvious difference in expression pattern. So what I saw is that the PDM3 gene was expressed entirely, and there's no spatial difference in expression pattern. So it's possible to hypothesize that there might be slight quantitative differences in the expression levels. And so there should be another question. So what kind of gene has a spatial difference in the expression? So I have a candidate for question 2. So a gene called TAN is the candidate to have the spatial difference. So TAN is the enzyme involved in the melanin synthesis. And so it converts a compound named NBAD to dopamine. Dopamine is the precursor of melanin. So it means that the TAN increases the precursor of melanin to promote the melanin synthesis. And so when it's overexpressed in the dorsal midline, so it will promote the formation of melanin. So, it's possible to predict that the TAN is highly upregulated in the posterior region of the dark females. So I tested this hypothesis by qPCR. So what I did is at first dissected out the pupal abdomen and chopped into 2 pieces, the posterior region and anterior region, and then extracted RNA. For the light flies, so what I did is I did the RNA extraction from the posterior region and compared the expression level of TAN between the segments and genotypes. And then as a result, so the TAN is highly regulated at the posterior region of dark females compared to, other genotypes or other segments. So when the segments were compared within the same genotype, the difference in the expression was significant. And so when the expression was compared between the genotypes, the expression difference was significant. So based on those results, so the hypothesis what I'm currently have is like this. So PDM3 expression might have the slight difference, and then it will promote the high expression of the TAN at the posterior abdomen of the dark females. And then it will create the phenotypic difference in light females and dark females. So to explain that hypothesis, currently what I have a model to explain that hypothesis. So it's abdominal pigmentation and Montim clade might be a threshold traits. So currently what I assume or what I predict is I predict that the PDM3 expression level can increase depending on the temperature. So there's no data to support it, but so currently I have this hypothesis. Well, because the PDM3 represses the melanin pigmentation, and at the higher temperature, the body color becomes lighter. So that's why the PDM3 expression can be higher at the higher temperature. And if there's any threshold, so it's possible that the PDM3 level is below the threshold. It's possible to have the dark pigmentation color. And if the PDM3 expression level goes up and if it's above the threshold, it's possible to repress TAN expression. So there's no data to support PDM3 represses TAN, but so based on the current data currently what I have, it's possible to predict. So this model can explain phenotypic plasticity. So how about the dominance? So I have the hypothesis. I have three hypotheses for this. So number one is for dark dominant species, and other two hypotheses are for light dominant species. So for hypothesis one, so if the light allele has the higher expression from the dark allele, and the allelic response to temperature is similar between alleles, the light homozygotes will have the highest expression, and the heterozygotes will be intermediate, and the dark homozygotes will have the lowest expression level. And if the threshold is always above the heterozygotes, regardless of temperature, so it's possible for heterozygotes to have the consistently dark phenotype regardless of temperature. So hypothesis two, so this hypothesis assumes that the allelic response to the temperature is same as dark dominant species. But so if the threshold is lower compared to the dark dominant species, it's possible for the expression level of the heterozygotes to span the threshold to create a discrete phenotype depending on temperature. And the hypothesis three assumes that the allelic response to temperature is different from dark dominant species. So in that case, the expression level has the steeper response to the temperature compared to dark dominant species. And in this case, it's also possible to explain why the phenotype can be discrete, because the expression level can span the threshold by having a steeper response to temperature. Okay, so let me summarize the former part of this presentation. So the results shows that the evolution of dominance and plasticity are directly connected. And the results suggest that the mechanistic connection between dominance and plasticity can exist, and it may be threshold-like property of development. So to test this hypothesis, so currently what I'm planning to do is the allele-specific expression analysis, qPCR, and CRISPR-Cas9 system to knock out the PDM3 gene. So, and the second one is interspecific dominance reversal due to the evolution of Fisher's modifier? So first of all, so why some alleles are dominant and others are recessive? So when the allele is newly born from mutation, so it tends to be recessive. And on the other hand, the wild type allele tends to be dominant. So to explain this, about 100 years ago, so Wright and Holden hypothesized that the dominance relationship between alleles is determined by inherent property of biological system. So it can be determined without selection. So let's assume the case, so let's assume an extreme case where newly born allele has the mutation not to produce any gene product. So when the new allele is aligned with the wild type in the heterozygotes, the amount of the genetic product will be half of the homozygotes with the wild type allele. So back then, so phenotype was considered to be determined by the presence or absence of the gene product. So that's why the heterozygotes can show the wild type phenotype, and then the newly born allele becomes recessive. So there can be exceptional cases. For example, when a newly born allele is the loss of function allele, and it produces loss of function protein products. And if the products must function by forming a tetramer, so in the heterozygotes, the most portion of the tetramer will lose the function, and then the phenotype will be the loss of function, and the newly born allele can be dominant. So in both cases, so the allelic dominance between alleles is determined by the process of the phenotypic determination. So it's the feature of biological system. And so it's the without selection. So for about dominance reversal between species, so there's a single experimental evidence. So it's about the color dimorphism is in lizard, white color and dark color. And in one species, the white color is dominant, and in another species, white color is recessive. So a gene responsible for the dimorphism was examined, and it turned out that the different mutation site at the coding region of the same gene was responsible. And then the different mutation site will create the differences in the protein function. And because of it, the dominance can be reversed. So while in this case, the difference of the dominance between species is determined only by the function of protein. So it means that dominance is determined by inherent property of biological system. And on the other hand, so there's an alternative hypothesis proposed by Fisher. This is also proposed 100 years ago. So he proposed that the dominance can be determined by the selection on the second side modifier. So when the new allele is born by mutation, so usually it tends to have a deleterious effect, and Fisher assumed that the newly born allele is co-dominant with the wild type existing allele. So in the heterozygotes, the deleterious effect should be reflected as a phenotype. It means that the mean fitness for the population will decrease. So it will be better, it's possible to conceal the deleterious effect from the newly born allele. And then, so what Fisher hypothesized is the evolution of modifier locus. So when the modifier locus evolves and it controls the allelic relationship between the primary locus, and then so it's possible to evolve to show the wild type phenotype. So and then the newly born allele becomes recessive. So in that stage, it's possible to conceal the deleterious effect from the newly born allele. So Fisher hypothesized that the dominance relationship can evolve over time. And so there are theoretical models to support the existence of Fisher's modifier. So one of the models is the evolution of modifier under strong frequency dependent selection. So they investigated many parameters, so let me simplify this. So when the frequency dependent intraspecific competition is stronger, the newly born modifier allele can be fixed in the population faster. And so they also examined another condition. So it's the effect of modifier locus on the primary locus. So if the effect is stronger, the newly born modifier allele can be fixed in the population faster. So there are many conditions or assumptions to support the existence of Fisher's modifier. However, so there's no experimental evidence to show the the dominance is reversed by the evolution of Fisher's modifier. So I investigated the existence of Fisher's modifier by using these three species. So one dark dominant species and one light dominant species, and this species is the sister species for these two, and it's monomorphic dark species. So actually they appeared in the previous part of my presentation, and those of the blue line, the dark dominant species shows the dominance of dark allele at the underworld of the temperature. And the Bocchetti, the light dominant species, it shows the dominance of dark allele at the lower temperature and the dominance of light allele at the higher temperature. And this species, Shobaka, shows the almost robust dark phenotype under all the developing temperature. So we investigated whether the interspecific differences of the genomic background outside of PDM3 affects the dominance relationship of the PDM3 locus. So at first, what we did is the crossing, so interspecific crosses. So Drosophila Shobaka dark strain and Bocchetti, this is light dominant species light strain. And it had the phenotypic plasticity in the F1 hybrid. So the dark allele was dominant under lower temperature and the light allele was dominant at the higher temperature. And so this phenotypic curve is really similar to the intraspecific cross in the Bocchetti light dominant species. And on the other hand, so when I crossed the Shobaka and blue light, the dark dominant species, and this is light strain. So they had the phenotypic plasticity at in the heterozygotes. So the dark allele was dominant under lower temperature, but they will be co-dominant at the higher temperature. And so when it's compared to the intraspecific crosses in the dark dominant species brulli, so the phenotypic curve was very different in the intraspecific crosses in the brulli. The dark allele is always dominant regardless of temperatures. So I focused on the combination of these two species to investigate the Fisher's modifier. And so what I did is to change the genomic background by introgression. So when the light strain of dark dominant species brulli is crossed with the Shobaka dark strain, it's possible to get the F1 hybrid females. And repeating the crossing with Shobaka dark strain and until the 8th generation. So it's possible to introgress the PDM3 light allele of brulli into the genomic background of Drosophila Shobaka. And it's possible to make the homozygous strain. And so for convenience, I call this strain as introlight strain. And then, so we compare these two crosses for this cross. So we're going to cross the Shobaka dark strain and the brulli dark strain. And the F1 females will have the combination of brulli light PDM3 allele and Shobaka dark PDM3 allele. And as a genomic background, so the 50% is from the brulli light strain and another 50% will be from Shobaka dark strain. And then if the modifier exists, the genotype at the modifier locus will be heterozygotes. And then, so in the next one, we cross the Shobaka dark strain with the intro light strain. And the progeny will have this genotype. So at the PDM3 locus, the combination of PDM3 allele is totally same as this cross. On the other hand, the genomic background will be very different. So it will be almost Drosophila shovaka. So if the modifier exists, the genotype of the modifier will be homozygotes. So it means that if these crosses have the different reaction norm, so the difference will be due to the genotype of modifier locus. So as a result, so at first, when the Shovaka dark strain is crossed just the blue light strain, so the phenotype had the phenotypic plasticity and dark allele was dominant at the lower temperature, and the two alleles were co-dominant at the higher temperature. So it shows the intermediate body color. So on the other hand, when I cross the Shovaka dark strain into light strain, so it still had the phenotypic plasticity, but at the higher temperature, the body color was much darker than this cross. So, well, it looks like the phenotypic curve is really different. So I tested the difference by two-way ANOVA. And so as a result, the effect of the genotype was highly significant. So it means that the phenotypic curve is very different between these two crosses, and the PDM3 allele is same between two crosses. So the difference in the reaction norm is due to the difference in the genomic background. So this result shows that the Fisher second modifier exists. So currently, I introduced the result by using this combination, the blue light PDM3 allele and the genomic background of the Drosophila shovaka. And now I'm preparing the many combinations of the PDM3 gene from many species, and for the background, I have many species of the many species. So these species are sister species. So it's possible so by using this system, it's possible to examine the evolutionary trajectory of Fisher's modifier. Okay, so let me summarize the latter part. So the Fisher's second modifier exists, and it alters the degree of thermal plasticity. And so this result suggests that the differences between dark dominant and light dominant species might be created by evolution of Fisher's second side modifiers. Thank you very much.
I'm wondering, so the last part you're talking about the finding the Fisher's modifier, and
Yeah.
you're saying it like for sure it's a single modifier. And I'm just wondering about the possibility that there are multiple transcription factor or other modifiers that might be in the whole, because you're crossing into the whole genetic background, right?
Yeah.
So just wondering like.
Yeah, it's possible. Yeah, it's possible to be multiple modifiers. So it's impossible to say this is monogenic effect. Yeah, I'm assuming multiple modifiers. Yeah, so if you are thinking about identifying the modifier, it can be difficult. Yeah, if the modifiers are multiple genes.
Multiple small effects, always fun to
Yeah.
find, right?
Yeah.
So does your idea, or does that connection between dominance and plasticity, how general do you think that statement is? For dominance situations?
So, I think it can be, it's possible to predict in another species, but I think it can be general because, for this case, so the phenotypic difference is very obvious. But in other systems, the phenotypic difference under different environment can be subtle. So it's possible to have the, well, difference in the dominance, for example, dominance degree depending on the difference in the environment by quantifying level. So in this case, it's really obvious by the eyesight. But in other cases, there might be just a quantifying level. So it means that I think for other systems, it requires to quantify the phenotype to, well, to test this idea. Yeah. So currently, I expect it can be general, but yeah, I think for other systems, it's not examined so far. Yeah.
I mean, maybe what makes a big difference here is not so much the overall phenotypic difference, but the effect size of a single gene, right?
Yeah.
So you're lucky that you're working on a Mendelian trait where a single locus pretty much controls the entire phenotypic difference. But if the phenotype is determined by 10 loci, then looking at the evolution of dominance in one of those 10, that I imagine could be much more difficult for something that's so clean and Mendelian.
Cool talk.
Oh, thank you, bro.
Yeah. I was wondering, and maybe I just missed it, but you were talking about this like threshold.
Yeah.
And it seems like maybe in this example that threshold was one gene producing pigment or some sort of enzyme that then triggered another gene, and that it was just sort of maybe like the quantity of something molecular that set that threshold, and that just like abundance of something allowed for another step to be triggered. And I wanted to know if I was understanding that correctly. I guess my bigger question is, what do you see as setting these thresholds? And do you think that that's generalizable across different mechanisms?
Okay, so you mean the generality of the threshold property?
Yeah, or like what is setting the threshold? And
Okay. So what sets the threshold, right?
Yeah.
Yeah, well, so actually the threshold-like response of gene expression is determining the relationship between the connection of the cis-regulatory elements and the protein of the upper gene. So wow, so it's due to the feature of the upper, it's feature of the protein and the feature of the cis-regulatory elements. For example, if the cis-regulatory elements have the multiple binding sites of the many binding sites with the upper protein, so upstream protein, it will have a steep threshold-like response. So wow, I think to determine the threshold thing is kind of it can be attributed to the feature of cis-regulatory elements or by number of binding sites. Yeah.
Thank you.
Thank you.
All right, let's all thank our speaker.
Yeah.